Hello friends, if you have not subscribed to my channel, then first subscribe and press the bell icon to see the latest video. In this video, let us talk about staining methods. As you know that the microorganisms normally present in the atmosphere. By using the staining methods, we identify their shape, size and arrangements. In this video, I will discuss about bacterial staining. So let's start the introduction about staining methods of bacteria. The staining methods are divided into two groups: simple staining and differential staining. Basically, the simple staining are used only for the identification of shape. For example, coccus, bacillus, spiral, comma shape, etc. On the other hand, the differential staining are divided into two different groups on the basis of bacterial staining and visualization of their different structures like flagella, nucleus staining, endospore staining and capsule staining, etc. The bacterial staining are divided into two different groups: gram staining and acid fast staining. To see this, you have one question in mind, that both are bacterial staining, but why divided into two different groups? Because the acid fast staining are used only for the Mycobacterium species. The Mycobacterium species like Mycobacterium tuberculosis possess both characteristics, either gram positive or gram negative. It cannot differentiate by gram staining. So this is the basic introduction about staining methods. Today's topic is gram staining. Gram staining is a part of differential staining that are used to identification of bacteria. Gram staining technique is developed by Hans Christian Gram in 1882 but published in 1884. By using this technique, the bacteria kingdom are divided into two broad categories, either gram positive or gram negative, using four different stains. These are crystal violet, gram iodine, decolorizer, and safenine. Those bacteria retain the crystal violet color and gives a purple color called gram positive, and those bacteria retain safenine and gives a pink color called gram negative. Requirements: 20 to 48 hours old bacterial culture, glass slide, dropper, blotting paper, microscopes, staining tray, inoculating loop, Bunsen burner, and gram staining kit. Gram staining contain crystal violet, gram iodine solution, and decolorizer and safenine. Now come to the procedure. Firstly, take a clean grease-free glass slide. Then, by using the dropper, put one drop of water or on clean glass slide. Then, with the help of inoculating loop, so this is the inoculating loop, take a small colony of bacterial culture and spread on clean glass slide to make a smear. Then allow to air dry and heat fix the smear. After the air dry, add 2 to 3 drop of crystal violet for 1 minute. Then wash with water. The mechanism of crystal violet is that the bacterial cell contact with crystal violet and retain crystal violet color and the smear becomes blue. Then wash with water for removing extra color. Then add 2 to 3 drop of gram iodine solution for 1 minute. The mechanism is that to contact bacterial cell, it form a complex which is not removed easily. Then wash with water. Then add 2 to 3 drop of decolorizer for 30 seconds. Decolorizer contain 95% of ethyl alcohol. In case of gram positive, do not remove color, and in case of gram negative, remove all color and smear becomes transparent. Then wash with water. Now add 2 to 3 drop of safenine for 1 minute. In case of gram negative, they retain safenine color and smear becomes pinkish color. Then allow the smear to air dry. Then examine the slide under the microscope. Observation. Observe the color Observe the color of bacteria and shape and arrangements under the microscope. Those bacteria gives purple color called gram positive bacteria, and those bacteria gives pink color called gram negative bacteria. These are the precautions. Always use fresh and young cultures. Do not overheat the slide during the heat fix, and smear should be thin. Thank you for watching this video.